Hello everyone, welcome to my YouTube channel. So this channel, it will be a study based channel which will be revolving around the field of plant pathology. And in this channel, both the theoretical as well as the practical aspects of plant pathology will be included. So I am hoping for a support from you. And I will start firstly with the most common and the basic topics of plant pathology. But if you have any kind of special videos or that you want me to make, or any specific topics that you want to learn in the field of plant pathology, you guys can comment down in the comment section. I will take your request into consideration, and I am hoping for a good support from you guys. I am a very passionate teacher, and I think I can make plant pathology easily understandable for you. So let's start for today with my first lecture, and that is the most easiest topic that has chosen in the field of plant pathology. So we'll be starting with the first, bacteria. All right, so as you can see here, basically if we define bacteria, so bacteria are defined as the prokaryotic organisms which are singular celled, means they are having a single cell, and they generally divide by means of a process known as fission. Right, the most important characteristic of bacteria being their prokaryotic nature. Right, so what do you understand by prokaryotic nature and eukaryotic nature? So prokaryotes basically is derived from two words, that is a pro and a karyon. So pro means primitive and karyon means a nucleus, right? So they will be having a primitive type of nucleus. Primitive type of nucleus means their nuclear material, that is their DNA, that is not being bounded by any nuclear membrane. As you can see here in the first picture, their genetic material, it is not bounded by any kind of membrane, right? However, it is different from eukaryotic cell, as you can see in the second picture, that the genetic material, that is the DNA or the nucleus, that is being enclosed within a nuclear membrane. So that is a major difference between a prokaryotic and eukaryotic cell, because eukaryotic means U means true, and carry again the carry-on, that means nucleus, right? So now if we have to talk about the lineages, there are basically three lineages. First is bacteria, that is your primitive type, that is your prokaryotic, which is also known as eubacteria or the true bacteria. Second lineage we have is archaea. Archaea means it is again a form of bacteria, that is known as archaebacteria, but they are the primitive type of bacteria, and they are different from your true bacteria in certain characteristics like their protein synthesis, presence of sterols, etc. So this we will discuss in the coming slides or in the coming topics, but for now you can remember bacteria and the eubacteria are one and the same thing, and they are different from archaebacteria, right? Then again the last next lineage, or a lineage which is separated from the bacteria, is eukaryota. That we will have is the plants, fungi, animals, etc. Okay, now this is the basic or a standard structure of a bacterial cell, where it will have a outer membrane. Now that outer membrane will have the three parts. First one is a capsule, which is the outermost part that basically act as a covering for your bacterial cell under the harsh environmental conditions, right? Then followed by that we have is a cell wall, then followed by that we have a plasma membrane. Now what are the main characteristics of its cell wall, how it is being formed, what is the role of plasma membrane, what is the role of capsule? These all we will discuss in the next coming videos. So for now you can remember it will have a envelope outside that will having the three parts. Now from that envelope there is another two important parts. First one is flagellum, which is your locomotory organ that helps in the movement of bacteria. So when it is singular, then it is known as flagellum, and when it is plural, then it is known as flagella, right? Then we again have the category of different types of flagella like monotrichous, lophotrichous, that we will all discuss later on, right? But the basic function of flagellum is locomotion, that means movement, right? And it is composed of a protein which is known as flagellin, right? Then again we have the second component, that is fimbria or pili. Now they are different from flagella in terms of their thickness and size, but the major difference is the type of protein they are being constituted. For example, your pili, that is composed of a protein known as pilin, and their major role, that is the major role of fimbria and pili, is in conjugation or attachment towards a particular surface, right? This is the envelope. Then inside this envelope we have is the cytoplasm that will have the genetic material of your bacterial cell, that is the DNA. Now since it is a prokaryotic cell, so it is not bound by any kind of membrane. Then it will have certain kind of storage granules and your most important protein synthesis synthesizing machinery, that is known as ribosomes, right? So it is a very generalized structure, and we can study the deep structure of bacteria with the each and important function of its cell in the coming lectures. Okay, now here you can see this is a picture that is showing the colonies of a bacterial under the laboratory conditions. So as we know that they are cultivated under laboratory conditions, that is they can be cultivated in vitro. So this is how a bacterial colony will looks like on a particular media, let us say on nutrient agar or under laboratory conditions, right? Now this picture basically detects a test which is known as ooze test, that is carried out in the field condition in order to detect the presence of bacterial pathogen in a plant. Now this basically represents a kind of bacterial streaming or bacterial moving out from a infected plant as it is very much visible in the second picture. So for this you have to basically cut your infected plant and you have to dip that infected plant into water, and you can see the milky ooze that is coming out from that cut end. That is because of the secretion of the, or that is because of the accumulation of polysaccharides from the bacteria, and because of which this has been there. And it is a very important and you can say a very easy and a portable detection method for bacterial pathogens, right? So this is all about this introductory lecture. I am hoping for a good response or for a quick response for you, and you can also comment down your videos that you want me to make, particularly the subjects that you want. And basically I am starting this series because I want to explore, I want to make you guys explore this universe of plant pathology, and it's a very fascinating science. And if you are preparing for your ARS exams, for your net exam, this is the best platform where you can have a better understanding of the concept, and I'll try to make all your doubts clear in a very much possible way, as much as I can. Okay, so thank you for it and have a good day.